Good afternoon, everyone. Thank you for joining us. As Sarah mentioned, my name is Mike Dombeck. I lead business development for Precision BioSciences. Precision is a genome editing company dedicated to improving life through cures for genetic diseases, overcoming cancer, and addressing health and wellness through improved food sources around the world. Our goal is to realize the full potential of genome editing. Our scientific co-inventors and co-founders are pioneers in the genome editing space. They both have over 20 years of experience starting their careers in the zinc finger space. Our technology features a customizable, designable interface with zero random off-targeting. Our patent estate has been through a patent challenge, and we have court-tested, documented freedom to operate in the gene editing space. We focus this technology on applications in genetic diseases, cancer, and food. We're enabled in our efforts through partnerships with companies such as Shire and DuPont, and validated by our investors, including Venbio and Fidelity. Over the history of the company, we've generated over $160 million in capital, which has now gone into full R&D capabilities, including 60,000 square feet at our facility in North Carolina, and a team of over 90 precisioneers at this point. That team is united in our common shared values for the company of respect, adaptability, perseverance, and innovation. And that team has brought to Precision the full range of capabilities to do product discovery, development, manufacturing, and to bring these products to market. The company is built around our core technology of ARCUS, the first therapeutic-grade genome editing platform. ARCUS is derived from naturally occurring homing endonucleases, endonucleases that have evolved in higher organisms for site-specific gene editing. In our efforts, ARCUS has been able to, we've designed ARCUS to be customizable to target whatever genes we wish to effect a break in, but have retained the features of homing endonucleases that drew our scientific inventors to them in the first place. These features include zero random off-targeting, the ability to fit an ARCUS nuclease into any delivery system, either viral or non-viral, the ability to customize and optimize these with exquisite control over both the affinity of the nuclease as well as the cleavage kinetics of the nuclease, a unique 3' overhang that is created when ARCUS makes a break, which allows us to pursue applications that demand efficient repair or insertions of DNA templates. And finally, the flexibility and adaptability in our design system to access any gene in the genome. We're applying the ARCUS technology across 3 business areas: genetic and infectious diseases, cancer immunotherapy, and food applications. In the genetic and infectious disease space, we are pursuing hepatitis B as an anti-infective, familial hypercholesterolemia, retinitis pigmentosa, and hemophilia A. Cancer immunotherapy, we're pursuing allogeneic CAR T with an initial program targeting CD19. And finally, in our health and wellness programs in food, our lead program is a low saturated fat canola oil. Talk a little bit about each of these programs. Starting with hepatitis B, there's a growing consensus that a hepatitis B cure is going to require an approach that reduces hepatitis B cccDNA in human hepatocytes. We have designed 2 ARCUS nucleases that recognize 2 different highly conserved sites in the HBV genome. We have tested these in primary human hepatocytes and demonstrated that these nucleases can eradicate HBV cccDNA both episomally as well as the integrated DNA, again, in primary human hepatocytes. For familial hypercholesterolemia, many of you are probably familiar with the PCSK9 target. A loss of function mutation in PCSK9 leads to very low levels of bad cholesterol, or LDL. Precision will be publishing data later this year in a large animal model where we have demonstrated safe, effective, and permanent loss of function mutation based on an ARCUS nuclease. Our Retinitis Pigmentosa program targets the P23H mutation, the autosomal dominant mutation of Retinitis Pigmentosa. We have taken this into the known mouse model for Retinitis Pigmentosa and the P23H mutation, and have demonstrated that treatment, subretinal delivery with Arcus is successful in rescuing photoreceptor morphology in mice. We will be taking these mice and doing visual acuity evaluations later this year. Finally, on the gene therapy element of our business, we're approaching hemophilia A with a unique approach. We're targeting the intron 22 inversion in hemophilia A and delivering an Arcus nuclease that targets a repeat upstream of the inversion. And what this does is it triggers the recombination event that originally caused the intron 22 inversion. What we're causing it to do is undergo a recombination event again that flips the inversion and restores the wild type conformation. So as Sandy mentioned this morning on the panel, if you heard, the goal of restoring wild type function with gene editing therapeutics is exactly what we're trying to do here in hemophilia A. And this program, we have demonstrated that we can effectively efficiently trigger this inversion in patient cells in vitro. We will be taking this into the hemophilia A dog model with a researcher at the University of North Carolina later this year. Moving on to our CAR T program, we are pursuing allogeneic CAR T, again CD19 as our lead program. Our current CMC process from a single donation, single leukopak from a single donor, is up to over 100 allogeneic doses that we can generate. Our approach to this is also unique in how we approach the CAR in the T cells. We use our Arcus nuclease to knock out the T cell receptor, the TCR locus, to create the allogeneic cell, but at the same time design the CAR sequence to match the break that we're creating in the TCR, so that the CAR sequence efficiently inserts directly into the break we've created in the TCR, essentially replacing the TCR with the CAR sequence. This gives us predictable levels of expression and therefore measurable potency and the ability to potentially dose titrate our CAR T therapeutic. Most importantly, again, this is allogeneic. You see this is a 12-day manufacturing process, but the point is this is an off-the-shelf product. So what we can do with this is have it on the shelf in the hospital and reduce the time from diagnosis of a patient to treatment down to a matter of hours versus the weeks that an autologous process currently takes. This allogeneic CD19 program has been through a mouse model. We've demonstrated that it does not trigger GVHD in an animal model. In addition, the data you see here with mice has been published in a peer-reviewed journal. We've shown the publication showed eradication out to 50 days. We have actually taken this mouse model out to 90 days and shown eradication at both a medium and high dose. As a complement to our therapeutic efforts, we also have our agriculture division, which is focused on improving health and wellness through more nutritious foods. Our approach here from a business perspective is to focus on partnership-driven crop improvements with large food companies that have known and demonstrated targets where applying genome editing techniques can greatly accelerate and improve the efficiency of generating a modified plant. Our lead program here, as I mentioned, is a canola oil program. This is being done in collaboration with a leading food industry partner, and the idea is to use our genome editing tool to shut down the genes used for saturate production in canola oil. Selected phenotypes of the canola oil plant will move into field trials next year, and the final product will be a healthy, low-saturated fat canola oil that is non-GMO. Reviewing some of the milestones for precision over the last couple of years, starting in the beginning of 2014, we settled the patent litigation we were in that demonstrably gave us the clear freedom to operate in the homing endonuclease space. Completed our Series A financing in the second quarter of 2015, followed a year later by our CAR T partnership, originally signed with Baxalta, now Shire, that brought $105 million up front into the company. Earlier this year, we published that mouse data demonstrating the durable elimination in the Raji tumor cell model with our allogeneic CAR T program. Also, about the same time, we hired a dedicated president of plant sciences, followed by a hiring of our chief development officer, who is seeing our programs through IND and into the clinic. And finally, we had our first interaction with the FDA just a month ago for a pre-IND meeting on our allogeneic CAR T, which should file IND in third quarter of next year. Looking at some of our upcoming milestones, before the end of the year, we are anticipating announcing a partnership with a leading food company, as well as publishing data showing durable, non-toxic editing in non-human primates for a therapeutic target. Early next year, 2018, we'll be opening our own dedicated greenhouse facilities for our agriculture division and anticipating announcing a gene therapy collaboration in the first half of next year. The second half of next year, we'll bring our own in-house GMP cell therapy and mRNA manufacturing facility, as well as I mentioned, the IND for our allogeneic CD19 program in the third quarter of next year. And then our lead gene therapy program is currently anticipated to file an IND in the first half of 2019. In summary, we've built precision to overcome life's greatest genetic challenges. We have built a team with deep expertise and commitment to what we're doing, and that enables us to make the best use of our platform, our ARCUS platform, what we believe is a true therapeutic-grade genome editing platform, focused on genetic disease, overcoming cancer, and generating health and wellness through more nutritious food products. We have built out the full discovery, research, development, and manufacturing capabilities to bring these products forward through development, and are executing on this through our partnerships and our investors. I am happy to take questions outside after this. Thank you again for listening to me tell you a little bit about precision.